Hello everyone, welcome to today's KAAPI with Curiosity. I'm Vijay Kumar, I'm a faculty at ICTS. I welcome you all on behalf of both ICTS and the Planetarium. As most of you know, the Planetarium and the ICTS have this collaboration on our outreach program. We've been running successfully from the past 3 years. We'll be finishing our third year anniversary this October. So it's nice to see many people coming regularly for this set of programs. So I've been asked to introduce both the Planetarium and the ICTS at the same time. So the Planetarium, in addition to its Sky Theatre shows, has a lot of informal science education programs, which are called REAP, Research Education Advancement Program. I myself was a student here 20 years ago and learned here. And so that's been the strength in developing a core set of people who can go on to scientific research careers. ICTS, on the other hand, we have a three-pronged mandate. We do scientific programs. We have in-house research teams working on different areas of science. We also have a strong and vigorous outreach program. The KAAPI talk today is a prime example of that. We also seek speakers for the KAAPI program from the ongoing scientific programs at ICTS. For example, the present speaker today is really visiting ICTS for a scientific workshop on mathematical modeling in disease and public health. So with that, let me turn to introducing today's speaker. Professor Carmen Molina-Paris is a mathematician, is a theoretical physicist. She did her PhD with Bryce Drevitt in the early 90s at the University of Texas, Austin. She did a couple of postdocs in the US and Spain, and then she moved back to UK. She's been at Leeds from the beginning part of this century, sometime there. She's a professor at the Applied Mathematics Group. Her research interests span things from elementary particle physics, quantum field theory, cosmology, and more recently and more vigorously into mathematical modeling of immunology. So she has her interest in trying to understand receptor-ligand binding in T cells, their interactions, signaling, and so on and so forth. I'm sure we'll have a fun talk, and I welcome Carmen to deliver her lecture. So before she begins, I would like to request Professor Saraswati Visheshwara to hand over a memento to the speaker on behalf of both ICTS.
Well, good afternoon, and I guess we're ready. So thank you very much for being here. It's really my honor to be here today. And before I start, I would like to say that feel free to interrupt with questions. I know we've got questions at the very end of the session, but at the same time, I think if you've got a question at the right time, it's more important to answer then than wait when maybe the question loses its relevance. Before I start, I'd also like to say why I've entitled the talk A Mathematical Adventure in Immunology. And as Vijay has explained, I'm a theoretical physicist and really moving into... You cannot hear me?
No, just right, don't.
What? The water is in the way, but I may need it. Can I? So since I'm a theoretical physicist, I'm not really a mathematician, even though I'm in applied math department. And immunology provided me sort of an adventure into mathematics and immunology. Also, before I go into the talk, I'd like to say that the part of the work at the end that I'll present is really the work of 3 PhD students, Maria Nowicka, Mark Day, and Jonty Carruthers. So I think it's important to acknowledge that young people with lots of curiosity come to our groups and do beautiful work that then it's my opportunity to present here today. So with this, before I start, I would like to play a video that was actually recorded by the Research Institute in Australia, the WeHi Research Institute. And with that, I think if we can start getting the curiosity back to the immune system, and after this little video, then I'll give my talk.
In 1960, Sir Frank Macfarlane Burnet was awarded the Nobel Prize for his contribution to the field of immunology. Burnet's clonal selection theory revolutionized our understanding of the immune system. To this day, Burnet's research remains the foundation of the way we think about disease and how the body responds to it. Clonal selection is the central mechanism by which your body fights infection. The following visualization presents clonal selection during an infection by strep throat bacteria. This patient has a strep bacterial infection causing a red, sore, pussy throat. The tiny round strep bacteria thrive on throat tissue. The bacteria are coated with hairs called M protein. M protein is what your body recognizes to be foreign. In response, your body will launch an immune attack against it. Your lymphatic network is an important part of the immune system, detecting any infections that arise in your body. The M protein hairs drain into the lymphatic system around this patient's neck. The lymph node is crammed full of B cells. B cells are guardians of your immune system, scanning the body for foreign material. Arriving at the lymph node are M protein hairs from the bacteria. Your body contains billions of B cells. Each B cell is unique and is sensitive to only one kind of foreign infection. By chance, one B cell is sensitive to the M protein hairs from the bacteria. This is the molecular surface of that B cell, magnified 10 million times. Its surface is covered with receptors that recognize M protein. The receptors grab onto the M protein hairs. This triggers an activation signal to travel into the cell. The activated B cell migrates and begins to replicate. In a few days, thousands of copies of the selected cell are made. This is the central idea in Burnett's clonal selection theory, where one cell is selected out of billions of B cells and identical clones have been created from it. Therefore, the cell is clonally selected. This creates an army of B cells that mass produce M protein antibodies and release them into the surrounding fluid. The antibodies circulate in the blood and travel into tissues. They bind to the M protein hairs on the strep bacteria. This flags them for destruction by the immune system. Other immune cells ingest the antibody-covered bacteria. They destroy the bacteria and eliminate the infection.
So after that little video, I will start my talk. And as I've said, please feel free to ask any questions at any stage. So I've actually had to think rather carefully about what I wanted to talk today, because this is pretty much out of our comfort zone. And I think I've learned even more than maybe you learned today. So at least it's been, for me, an adventure already. So what is it that I would like to tell you today? I've structured the talk to really go back to history. History for a number of reasons, as you'll see, and perspective. Disease, it's not just the infection, but also the social understanding that might sometimes, or most times, not be scientific understanding of what disease is. A brief introduction to immunology so that everybody here gets to know and to learn more about what our immune system is. And then I thought it's important to see how recently new experimental methods have allowed us to really answer some of the questions that have been waiting to be answered. And I will conclude with 3 little examples of how mathematical approaches can help provide maybe some answers. And I'll conclude with some looking forward of what is maybe some of the pressing questions. And of course, one always has to say thank you at the end. So what is the perspective and the history? I guess we all know that disease is not something we like, right? Nobody likes to be sick, unless you want to be in bed all time and, you know, your mother or your father bringing warm milk with some turmeric and honey or something. But we don't really like being sick. In order to prepare for this talk, I came across this book entitled A History of Disease in Ancient Time by Philip Norrie, who's Australian, I believe, written in 2016. And what he claims, of course, with facts, is that disease has killed more people than put together wars, famines, or other disasters, which is quite a big claim to make. But I think important is to really put in the context that disease has also, or is also associated with people moving from one place to another, either by invasions, sometimes not that peaceful, like soldiers and armies, and sometimes just exchange of goods with trades. And these epidemics have caused changes throughout the course of history. So this timeline, and every time I put this little box in red is the source, so if anybody's interested in finding where I got these figures from, you can see, for example, that, well, 200,000 years ago, that's when we all emerged from Africa, and different stages of migrations, different years. But for example, 2,000 years ago, which is not that long, we had lots of cholera, and that was also linked to the trade in the Silk Road, which is, you know, pretty close to this part of the world. So humans, pretty much since we started as a human, had to survive to either fight or avoid infectious diseases. So for example, when you're little, or at least maybe now, right, we've got vaccines, and I'll talk about vaccines later, but if you fell and, you know, you cut yourself or grazed with some piece of metal, tetanus was a big concern. When you were little, diseases like diphtheria or measles can be really rather nasty, and in fact, right now there's a not great outbreak of measles around New York city area. Food or water contaminated also is a major problem, and infections like leprosy, tuberculosis, or malaria, for example, and 2 big killers throughout our history, smallpox and the bubonic plague. So for example, this other timeline, I quite liked it. It's much more recent, so it's only 5,000 years BCE. But what you can see is that plague has been, you know, not just the ones that we remember because we've learned them in history, but even very early on in Greek Roman times and even before, plague and smallpox have been really major killers. And just to provide, I would say, some examples I put this slide because sometimes, I don't know, maybe one associates infections and disease with poor people, but even Alexander the Great, in fact, seems to have died either malaria or West Nile virus infection. And also, disease helped him conquer Greece. There's also evidence that Ramses V, around 1,000 years BCE, died of smallpox. The mummy has marks on the face from smallpox. And as I was mentioning, these emerging infectious diseases are associated with human modification of the environment as early on as we can imagine things that are considered advances, like settling down, having domesticated animals, agriculture, allowing for more people to be supported in a community, that by itself led to the emerging of new diseases. And something I really didn't know before preparing for this talk is that measles very likely was adapted to humans in Mesopotamia about 5,000 years ago from a dog virus. So the domestication of dogs and having dogs close to us, that virus adapted to humans. And I thought it would be interesting for you to hear when, if you didn't know, that in the Indus civilization, 2500 BCE, that's when measles came to India. So, you know, I guess we've been suffering these creatures for a long time. I thought I'd bring this other example for the particular reason that, as I was saying, maybe one associates infections with poverty, so that's not necessarily the case by a long shot, but also with medicine itself. So this example is Ignaz Semmelweis. He was a Hungarian medical doctor who decided to count mortality in women delivering babies. And he was, in fact, he was in Vienna in a hospital. He was Hungarian, but of course at that time he was in Vienna. And what he observed is that children delivered by medical doctors or trainees, sorry, not the children delivered, the mothers, after delivering the baby, would die with a rate of 15%, which is rather large, whereas those women who had had the babies by very natural methods with a midwife, that mortality rate was only 2%. He actually made an empirical observation, and with that, he presented that to his colleagues. At the time, I think it's important, and this is what I had mentioned earlier, this how society or the understanding of disease sometimes doesn't agree with the scientific understanding. At that time, so we're talking about earlier in the 19th century, the germ theory of disease, the fact that disease comes from a germ, a bacteria or a pathogen or a virus, was not known. And what was believed at the time, it was something called miasmas, like bad air, this bad air floating around, and that was infection. So his empirical observations were that increased mortality was caused by the infected hands of medical doctors. You can imagine, I mean, maybe there's a doctor here, but you can imagine at the time someone basically saying to a doctor, "Excuse me, sir, but you're the cause of this infection." This gentleman didn't like that at all. So they ostracized him to some degree that he left Vienna. He went back to Pest, and he started implementing doctors before the procedure to wash their hands in some chlorinated lime solution. That brought hospital mortality rates in Pest to 2%, whereas in Vienna and in Prague, you know, these doctors were being a bit of a, you know, stubborn medical doctors. They didn't want to wash their hands because, of course, that delays, and why would you do that? And who is this guy to tell me what I'm going to? So that was that. And then another example that I was quite pleased to see, I mean, and it's remarkable what you can find. You can find this report written in 1913 from a major, a British major who was posted here in India, because at the time, they were going to open the Panama Canal. And of course, you would think, God, India is here, Panama is where it is, why should we worry? Well, he was rather intelligent, and I think he thought very carefully. So in 1913, yellow fever virus was still not in India. And he thought, well, if I look at this country, it has basically all the properties so that if this virus gets here, you've got the grounds, given the temperature, given the mosquitoes. Everybody knows that by the time they knew that mosquitoes carried disease. So he thought this would be fertile ground for something like this. And at that time, they already knew it was endemic in Southern America. So if it's endemic in South America, you open the Panama Canal with the trade, with the commercialization, boats bringing things, we're going to get it. It's going to come to India. So already in 1913, and as I said, in this lovely piece of read, Sanitary Awakening in India, written again in 1913, you can read it all about. He made some recommendations, so he really knew what it took. So what he was thinking is, please appoint medical officers at the ports in South America, Hong Kong and Singapore, when you're coming en route, investigate at the time in India where these mosquitoes might be, and try to reduce breeding places. So already in 1913, there were people with that understanding. So with this background, I think I've convinced you that we need to understand how our immune system might fight infections so that we're protected. Something that was observed very early on was this idea that there is heterogeneity in the responses in a human population to disease. What do I mean by heterogeneity? What we mean is some people don't even know they're infected, like this individual. Some people, a bit of fever, a few days in bed, nothing major, and some others, either they die or they really don't handle the disease well at all. So what is the immune system? I thought, because there's all these words one can use, and I thought maybe a definition of the immune system is, just imagine, you've got your house and someone knocks on the door. So how willing are you, right? If you don't know who's knocking on the door and going to let them in, you want to sort of think twice. So that's exactly what the immune system is doing. If I've got my little virus, and this is sort of the barrier of my skin, there is all this army of cells and molecules that I'll guide you through them to protect us from these pathogens. Is that clear? So protection from the different, and we'll describe also what different is in the immune context. So how is the immune system then organized? It's organized by 3 different levels of protection. And my definition of immune system for today is a collection, and this is very important, and you've seen that in the video, so that's why I wanted you to see the video. These are extremely motile cells. Immune cells are extremely motile. Cells in your lung stay there. Tissue cells stay where they belong and form the tissue. They don't move around the body. Any cell of the immune system is extremely motile. That's one property. They get into tissues, they get out of tissues, they get into blood, they get into the lymph nodes, they travel in the lymph. And lots of very important and specific molecules. They will protect us. I've been talking about pathogens, but I think here we also need to reflect on the fact that the immune system is a very important arm of protection against cancer and tumors. I'll talk about that too. The human immune system, as I was saying, 3 levels of protection. Anatomic and physiological barriers, like our skin, even the nostrils. I mean, I remember when I was little and they told me this, I thought, how silly. The nostrils, the little hairs in your nostrils, that's also part of the protection, because things get stuck there so that they don't go in. Innate immunity and adaptive immunity. I'll explore all this, but important to say that adaptive immunity, the 3rd layer of protection, is only for vertebrates. Most organisms on our planet, they survive just with some physical barriers and the innate immune system. So I thought I'll show you this picture just to explore a little bit more. So here, as I was saying, you've got barriers, which can be your skin. And of course, your barrier is your intact skin. As soon as you've got a cat, it can get infected. We all know that. That's bad news to have cats. And then, of course, you have a low pH in your stomach. Then the other 2 barriers are innate and adaptive. I've liked this picture for 2 reasons. One is because it really is captivating. It gives you everything in a nutshell, but also because you see these boundaries are a bit zigzaggy, like a little puzzle piece. There is not a clear definition of what is innate and what is adaptive in some sense, and I'll explain because, of course, both systems talk to each other. But in every part, either the innate immunity or adaptive immunity, you always have together a set of specialized cells and sets of special molecules that support the activity of these cells. Some of these molecules are secreted by immune cells, others are part of the inflammatory environment that goes with infection. So when we refer to humoral, if you've not heard that word before, humoral refers to something soluble, like a molecule, and cellular, a cell. So what are the cells that we'll talk about? Oh yes, and then you've seen the antibody. So the antibody is a soluble molecule secreted by some cells. And also, if you've never heard the word lymphocyte, lymphocyte is equivalent to white blood cell, T cells, and B cells for today's talk. So to give you a background to these cells, we don't need to name them all, but in the innate immune system, I will talk about these two dendritic cells and macrophages because they are cells of the innate immune system that talk to the adaptive immune system. And in the adaptive immune system, we've got B cells. I will not say any more than the fact that they make antibodies, as you've seen in the video. There are T cells of two types, helper cells and cytotoxic. I will say a little bit about helper cells, but for the rest of the talk, we'll be zooming into CD8 T cells, the cells that actually kill infected cells. So something very important, so now let's just go through what I would like to say is to keep in mind the differences between innate and adaptive immunity, and one can understand in light of evolution that it took the transition to vertebrates to build up the complexity of what adaptive immunity is. Furthermore, that always comes with a cost because it's a very expensive system. So if we look at timescales of immune responses, innate immune responses are almost immediate. Just imagine, you ever been cooking and you cut yourself with a knife and immediately it becomes inflamed a little bit, red, or if you burned yourself, so almost immediate, that's inflammation from your innate immune system, few immediate or a few hours. Adaptive immune responses take days because they are specific. I'll get to that. So again, for pathogen specificity, innate responses are the same no matter what the pathogen is. Bacteria, fungus, virus, whatever, they are the same. On the other hand, and that is the extreme advantage of the adaptive immune system, these responses are specific. It is going to be different if it's malaria or flu. It might even be different if it's flu of strain 1 or a different strain. So it really is suited to the molecular details of the pathogen. And memory, this is very important, and this will move us into the next thing I want to say, which is vaccines. Memory, what is memory? So just imagine, memory is nothing else but if I am infected now and I recover, and I'm infected in the future, will I become ill? No, I can be protected. So in the context of protection in the future, if I've been exposed to the pathogen a first time, which is called the primary infection or primary response, the innate immune system has no memory, no recollection of whether or not you've seen this pathogen one time or 1,000,000 times. On the other hand, the adaptive immune system will remember and will explain how that is built. And this memory, everybody probably, and I hope, everybody here has been vaccinated. So that is the link. And what is the link to provide both memory and specificity? Two cells, or two cell types of innate immunity called dendritic cells and macrophages, and I always like to call them vacuum cleaners. These cells do, and you saw them today, clearing the infection. So they go around the body inspecting for dirty things. I mean, let's just call them dirty, but it can just be, things that, we are not. And as I said, they provide the link for the specificity required for your adaptive immune responses, so macrophages and dendritic cells. So how do they do that? So this is the surface of a cell, a dendritic cell or a macrophage, and this is the inside. So you can have a virus that's gone in or a virus that's going in through like a little bacteria like you saw today in the video. What the cells are doing is once they vacuum clean, they actually sort and process what they've seen. And what they do, so all this is like some machinery, so virus goes into these vacuum cleaners, some machinery binds to another molecule called MHC. Let's not worry about what MHC is, just a very important molecule. And then you see how you get the MHC molecule with a little bit on top. That little square there is a peptide. A peptide is a fragment of a protein. So it's a protein that comes from whatever they've engulfed. Most times they engulf debris, so it's our own proteins. But when they've engulfed a virus or a pathogen, they are putting a little flag saying, this is what I've caught. So these dendritic cells and macrophages bring the specificity, even though they are innate, in the bringing in, processing, sorting, and presenting on the membrane. They will talk to your adaptive immune cells. As we've mentioned them, B cells or T cells. But before, yes, sir. Yes, on the previous slide. I can't hear you. Yeah, or if you get up and then everybody at the back could hear you too. Yeah. Well, I have to make a disclaimer. Well, the Golgi apparatus, it has to do with protein, but lots of trafficking. And I have to say, I'm not an expert in the immunology of presentation. But it's just trafficking. It's general trafficking, presenting, processing of protein. But I can give you lots of papers to read. Yeah. I mean, as I said, oops, we've moved. I'm a physicist, so I'm really an imposter here. You know, no mathematician, no immunology. So, yes?
I have a question as well,
Sure.
again in the previous slide.
Yes?
So you mentioned here that this macrophage engulfs, and then if it's some kind of foreign, a virus or something, there's a protein that's expressed, right? So is it always the same protein?
Very good question, very good question. It's not always the same protein. Oops, actually, is there chalk there? So I can do a little detour. So very important question. It's really zooming into detail, so I don't want to lose our audience. But the question is it always the same thing? No. So a protein is a very long sequence. Yeah? Thank you. So a protein is a very long sequence of amino acid residues, right? The ones that are presented, those little squares there, are only 9 units long, 9 amino acids long. Yep, so 9 amino acids. So they're really short. So you can imagine, haven't you ever do that? If someone gives you a long piece of ribbon and you have to cut in small little pieces, so you can cut there, but you can also cut like that. So that is what it's called. All these little peptides are called epitopes. So you can imagine how many collections you can do if you've got a long protein to make 9 mares. So no, they don't always present the same. And sometimes what happens if they present, if the one you present is one, I mean, let me give you that answer in the next slide so that you understand. Yeah? Oh, there's another question at the back? No, okay, fine. So antigen presentation, this is called antigen presentation. You vacuum clean something inside, chop it up, and present bits of it. So before I continue with immunology, I would like to really reflect on the relevance of adaptive immune cellular T-cell responses and why. And this links to vaccines. So I was going to call it a milkmaid story, and then I read this other article. So maybe it's not a milkmaid story. Everybody has heard of vaccinations. Everybody has heard of variolation. Do you know what this picture is depicting? So, of course, it's a very baroque, right, exaggerated, some medical doctor putting something in this little child's arm being held by a very devoted mother, which I have to say at the time, you know, not a lot of people would volunteer to do these things. But the fact of the matter is, what did we understand? Remember how I mentioned that some people survived infections. So even, 430 BC, which is a long time ago, there was an amazing, he was a historian, but he was also in the military. I was just reading yesterday, I never know how to pronounce it properly, Thucydides, something like that. What he observed was those that survived the plague would not suffer it a second time. That already tells you that in his lifetime, he saw how many waves of plague, right? Because if he saw people surviving and being infected again. So already, the Chinese, 1,000 years ago, were doing variolation. What is variolation? So if you get smallpox, you get little pustules with bits of pus, white little things. So what you would do is you take some of that and basically go to your best friend who trusts you and scratch a bit of, it's true, you scratch a little bit of skin and you transfer your liquid. As you can imagine, this is extremely, this is being very vulnerable to fluids. But this oozing matter from the source of smallpox have something that might protect you. And that was observed. So I guess some people realize, well, if you've survived and you're protected, what is in there? So, lots of things that might be considered very unethical now were being done not that long ago. So the Chinese, as I said, were doing this a long time ago. So even though, have you heard of Edward Jenner? Oh, very good. So if you've heard of Edward Jenner, I always read that he had seen this milkmaid who was milking the cows, and because, of course, cows get cowpox, which is similar but different from smallpox, since she was milking the cows, he observed that the milkmaid wasn't getting smallpox. Apparently, that is not the true story. The true story is that there was a previous medical doctor called John Fuster, and if you look at the history, the numbers match, so Edward Jenner was small when this doctor had his clinic, and of course, they knew that individuals that were exposed to cowpox were protected to smallpox. And he had a little hospital. So it's apparently the case that Edward Jenner was aware of these practices. So in 1796, no milkmaid myth, he did this procedure to this baby called James Phipps, and he survived. I really would like to stress today, vaccines have eliminated or minimized devastating diseases, and I cannot emphasize what difference it's made. And it's, for me, very sad to see that polio that was almost eradicated, it's coming back just because of war, in this case, or measles, when we've got a vaccine, just because some people, entitled, as I might call them, decide not to vaccinate. And herd immunity is then gone. Because in order for vaccination to work, most of us should be vaccinated. There are people who may have certain conditions like autoimmunity, and of course, vaccination is not appropriate for them for very good reasons, but immunity at the level of vaccination, this protection is at the level of the population, so we shouldn't be entitled. That's my, yes, oops, two. Yes, oh.
Can you just explain properly what herd immunity means?
So that's exactly what I've said, that if sufficient numbers of individuals are vaccinated, so how do you continue the epidemic? By transmission. So if enough people are vaccinated, there's no transmission. So it's almost like hiding. If you have enough people, you can hide an individual from being caught by the police, right? If enough of you, protect your friend from something, they won't be caught. So that's it, yeah. If one person is not vaccinated, and this happens, there are people who cannot be vaccinated because they've got autoimmune conditions, and vaccination for them would be death. So they are protected from measles because all of us have had the vaccine, and therefore we don't transmit the disease.
Okay, but does that, So it's basically about the chance of not
Exactly.
meeting a person who has measles.
Sure, it's a matter of contacts and probabilities, of course. Yeah. But that's like everything, right? It's a matter of numbers.
Can you infer from this, your study, that keeping contact with animals can protect you from disease, building antibodies?
Oh, well. So that's a very good question. So it's two things. I mean, what we know is in some instances, if two pathogens are very similar, and one pathogen, so for example, what I know? Because, I mean, this is not my study and I'm not an immunologist. So the evidence here is cowpox is sufficiently similar to a human disease. So let's just define it very properly. Cowpox is an animal disease, not a human disease. Smallpox is a human disease, not a cow disease. But they're rather similar, and it's to do with these epitopes that I was saying. So if you get the other one, and I'll explain how you build that memory, when smallpox comes to you, you have antibodies. You've got an adaptive immune memory. Yes? Animals and contacts, we're seeing it now. Avian flu, lots of contacts will actually make that adaptation. So absolutely. So it's good and bad to be together.
Paradox.
Life, isn't it just too much, too little? Yes. Can you speak up, because I don't think others will hear your question.
So I read this scenario, and there are some movies based on it as well. That due to global warming, a lot of pathogens which have so far been dormant are going to become active. And
Did you say active or inactive?
active. So, for example, there was this movie, I forget, with Val Kilmer in it, and it said they're researching in an Arctic station, and they unearthed the fossil of basically unearthed the fossil of an extinct animal, and that when it starts to defrost, there are pathogens which are hidden inside the body of that particular animal, which starts to become active, and then the entire team dies. That pathogen then starts spreading from the Arctic station into the global community. So such a scenario is possible, right?
Well, I mean, who am I to say if such scenario is possible? I can tell you the following. We are just going to start, in fact, in September. One of our PhD students will start working on Yersinia pestis, which is the bacteria causing the plague. And some bacteria can be in latent states. I have no idea how long this latency period can be. But it's almost like an inactivated form. It's almost like these spores that, they are inactivated and they need a bit of activation. So I could imagine that such scenario is possible. There is, in fact, one virus called Marburg virus. It came to Germany and it caused a terrible outbreak because someone brought the skin of dead monkeys. And there was just some little bits of viruses, dead viruses. So that can be, I mean, we know, for example, bacteria are made from inactivated bits of viruses. So I could imagine something, but I don't know the film, and I can't predict these things. So back to what we were saying. So we've understood vaccines.
Sorry, one more. These
I
9
can't see.
peptides.
Who's asking? I can't see. Yeah.
The 9 peptides, so they are the markers of a particular antigen.
Say that again. Sorry, V.
The 9 peptide sequences, I mean, the sequence of 9 peptides,
Yes.
they are the markers of that particular antigen.
Yes. I can't understand the first word you're saying. Can you repeat that again?
The peptides,
The peptides, yes.
the 9 peptides, the sequence. So all of those 9 peptides are necessary, right, to distinguish one antigen from the other.
I'll explain in a minute. T cells can only see, so in this picture, this little red box needs to be of a certain length, and it's usually 9, 10, 11, 12.
So these are basically, it's a sequence.
Yeah, but they're very promiscuous and cross-reactive. So I'll explain that in a minute. It's not lock and key, but yes, it cannot be longer. It cannot be 17. It cannot be 5.
So it's 9 peptides, basically.
9, 10, 11.
So
Yeah.
they are what distinguishes one peptide from one antigen from the other.
Absolutely. Yeah, that sequence. Thank you. So now that we go back to immunology, I'll finish with this slide to say, and that's all I'm going to say, B cells make antibodies, and this is one section of a lymph node, one specific region called the germinal center, where all these interactions between all these cells takes place. And then, as we were saying, what is the relevance of T cells? And this is where I think we need to stop to understand. So imagine this is our dendritic cell or our macrophage, our antigen-presenting cell. It can talk to two types of T cells, CD4, the ones that provide help, and CD8s, the ones that can kill infected cells. And what you've seen before, which I was saying, that little square has become now a round, so the red square has become now a green circle. So your antigen-presenting cell is presenting on its surface an MHC molecule with the bit of the peptide, this part, we're zooming in, and it talks to the T cell receptor on the T cell, whether it is a killing cell or whether it's a helper cell. So the specificity is related to the interaction between the T cell receptor and the bit of the peptide presented by the antigen-presenting cell. So that's the relevance of this interaction and why antigen-presenting cells, such as dendritic cells and macrophages, provide the link to bring us to the specificity required in your immune response. So as I was saying, CD4 T cells help B cells make antibody. So part of the steps to make antibodies, these B cells need to see a CD4 T cell. That was not explained in the video, but just so that you know. And remember, antibodies are important because they attach themselves to pathogens to mark them. It's almost like giving them a flag that's going to make them recognizable. And then I'll finish this immunology bit with saying that CD8 T cells, they are cytotoxic. Cytotoxic means that they can kill. And the way they kill is if I've got this bit is the cytotoxic T cell, the cytotoxic cell, and this is the infected cell. If a cell is infected, as we know, it will express this bit of peptide, it will be recognized, and if that interaction between the two molecules, the T cell receptor and the peptide bound to the MHC, these cells have little containers inside of them with some molecules called granzymes and perforin. So if you hear the word perforin, it makes you think of perfor, which is to make a hole. So these cells are very nasty in the sense that they've got a little box of very potent molecules that if thrown into the intracellular space will make this, and they actually shoot them with exquisite precision, pretty close, because of course they don't want to be damaged themselves, and so very close to the membrane of the other cell. And of course, if a cell has a damaged membrane, it doesn't maintain its osmotic pressure, and it will die. Is that clear? So for right now, we'll just finish with the immunology of CD8 T cell responses, and the important thing is here. In the same way as in the video, you saw the specificity of a B cell response. The specificity in the T cell response is as follows. Only about 1 cell in a million in your lymph node will be specific for the particular epitope that comes, or the particular antigen that comes, the peptide that comes from your pathogen. When that response starts, remember the clonal selection, you've got this clonal selection. Clonal selection means you select those that are specific for the pathogen. You have an expansion, lots of cell divisions, and the colors here mean that you start generating cells with different functions. Some of them, the blue ones, will be responsible for killing infected cells. The red-orange ones are what we call memory. So in this way, in this in adaptive T-cell response, you generate two types of cells, the ones that are going to kill and the ones that will stay later to protect us from a secondary infection. Yes? Yeah.
Just an observation rather than a question. I see the days post-infection is kind of a logarithmic scale. It's not a linear one.
Well,
Is that because of cell division process, or is there a particular reason for that to be
So
logarithmic?
what you have is around day 5, day 7, you've got this big increase in cell numbers. By 2 weeks, you've already cleared antigen. You've already cleared the virus if it's an acute response, yeah. And then you just go into what is called the homeostasis, which is keeping these cells alive for later purposes. Yeah. But I mean, this is not data. This is really, like I could have gone with my pencil and draw it. Order of magnitude. Yes?
Talking about infections due to vaccines or actual diseases, viral diseases. When you
What I
treat
do?
treat a person with a vaccine, injected
Oh.
virus, and this is the reaction, or when a person is infected.
Well,
The memory development, this is due to a vaccine? Or
Well, exactly. Well, this is both, because when they give you a vaccine, you have a small immune response, but you do. Yes, the good news is it's sufficiently mild that you don't have lots of fever. But I can tell you, when my first child had his first vaccine, he was very lethargic and with high temperature for a few hours. And I was, in as much as I truly believe you can start, but this is what you see, how you can start doubting yourself when it touches you, right? When it touches others, we can be, for you, not for me. But no, this absolutely, an immune response to any epitope, whether it's an attenuated, so yes, very good question. Let me continue in the interest of time, because I've realized it's almost 5 already. So just for the big picture of vaccines and memory, first infection, the virus is allowed to grow a lot. You mount an immune response. In weeks, for years, you keep your memory cells so that when you've got a second infection, you combat it very quickly. You increase the number of memory cells. Important is memory cells fight infection much better than naive cells. Yeah, so that's another thing. Well, with this, I'll move on to how can we now measure experimentally immune responses in this time and age. So I thought it's always good to reflect, because we're here now because someone else was somewhere else before and they've thought about it. So what is it? I've been talking about these B cells and T cells, white blood cells, lymphocytes. I think that almost shocked me. I couldn't believe it. In 1674, we already had visualized with a microscope, a Dutch scientist, red blood cells. He, in fact, saw white blood cells, but he didn't distinguish them as different, apparently. Because, of course, nobody knew about them. So, this is the 17th century. They knew about red blood cells, but nobody knew about these other cells. Only 200 years later in science, it's a long time, that's when they identified white blood cells. And only, early in our previous century, that's when they started to be classified. And as I've said, white blood cells is equivalent to lymphocytes, B cells, or T cells. I thought I'll add this for 2 reasons. The first one is there is an incredible immunologist, Goans. He was an incredible scientist. He writes in this lovely paper in '96 that he had been in Paris. There's a wonderful research laboratory of infections, Pasteur Institute, of course, Pasteur. So he comes back to Oxford and he writes very humbly, I came back in 1953 with little idea of what to do next. And we're talking about a brilliant scientist. For me, that is very inspiring. There was a very eminent professor of pathology at Oxford who said, these lymphocytes, why don't you work on them? We don't understand them. And he quoted another eminent scientist that this ignorance of not knowing what the cells are doing is the most humiliating and disgraceful gap in medical knowledge. You've seen the video, and that's why I played it earlier. Burnett in 1959 had postulated this theory of clonal selection before the lymphocyte had been identified, which is very interesting. This is where very bright people have the right ideas, but the experimental methods don't allow you to support them. Like, for example, we were talking about the medical Hungarian doctor. Nobody believed him about washing the hands, right? Because there was no evidence. In 1962, Goans and someone in his group put everything together, and they could say that this small lymphocyte that had been observed earlier is the antigen-specific type of cell that will do the job required for the clonal selection of Burnett. So you can see how it's important to work in collaboration, because we don't all have the complete picture. Should be fools to believe. How can we measure cells? Well, there is a beautiful technique called flow cytometry that actually makes use of antibodies. So antibodies, as you have seen, can bind very well. So you can make antibodies to lots of different molecules, and some of these molecules are on the surface of T cells and B cells. So it's almost like decorating yourself, like putting flags on your skin, and then you put these cells through this little tube one at a time. You shine a laser, and of course, the antibodies are labeled with fluorescent tags, and you can see them in colors. So it's color, multidimensional color sets, as you can see here. So here is for 2 labels. So you can say, well, if my cell has this label, it's a CD8. If it has this other label, it's a CD4. It's almost like this, like imagine we were forced, if you're from Spain, you have to wear a shirt of the Spanish flag. And then they pass you through these things. And then you know, she's wearing the Spanish flag. Then, another beautiful piece of experimental methodology is at the single cell level, understanding what is inside a cell by RNA sequencing. And as I've mentioned, flow cytometry allows you to look outside the cell, sometimes also inside, but for the most part is markers, proteins on the surface. With sequencing, you can look inside at the single cell level. What is one single cell machinery? Proteins expressed, lots of things. So combined, they can give you a very good understanding of how different cells are in different stages of the immune response. And then I've chosen, there are lots of technologies, I'm not going to go through all of them. One I know a little bit is 2-photon microscopy. And why has this been important? Because usually, if you either do in vitro studies, that's in your little dish, it's not in vivo. Even when you do in vivo and you do animal work, with the ethical permissions, of course, you sacrifice the animals and do histology sections of the tissue, sections of the lymph node. Those are static pictures. You take a photo at a given time, but you don't have the evolution in space and time. 2-photon microscopy allows you for about 30 to 60 minutes to visualize in vivo an immune response. And I'll talk about that when I move to the mathematical section. So 2-photon microscopy has allowed, of course, you have to label the cells with fluorescent dye so that they can be seen. And here you can see, it's hard to read, I'm sorry for that. This part in red and green are antigen-presenting cells and T cells in a lymph node. And you see whether the uninfected scenario looks different from the infection at different times. And here what you can actually quantify is trajectories of individual cells in real time in a lymph node. To me, that is remarkable. Yes, so this is happening now. So now we move to the mathematical adventure. I'll start with what I've called the first problem. The first problem is, we've talked about these T cells, but where do they come from? And that is, to me, a beautiful miracle of nature and evolution. There's an organ called the thymus, somewhere above the heart, and this is where stem cells, hematopoietic cells from the bone marrow move to the thymus, and some of them will become T cells, not all of them. So the idea is that some stem cells come into here, they do a journey, they are exposed to an environment and signals, and some of them will become T cells. So why is this important? Because you can imagine, and if you have any problems with this thymic development, as it's called, you're not going to have a population of T cells that are going to protect you and provide adaptive cellular immune responses. So what is it that we know? So as I've said, our T cells, CD4 and CD8, come from hematopoietic cells from the bone marrow, progenitors, exactly, that in order to complete the maturation, they move from the bone marrow to the thymus, somewhere above the heart. And important, and we've talked about these peptides that are on antigen-presenting cells talking to the T cells, exactly that exam is what happens in the thymus. These precursors of the T cells have to talk to antigen-presenting cells, so round circles, precursors T cells, dendritic cells with this structure, antigen-presenting cells. Those interactions will determine whether or not your precursor T cell will migrate to the blood and join the peripheral pool of naive T cell. How does that happen? So it's a very synchronized and very well orchestrated journey. They have to follow a certain route. And as I've said, in these transitions, you're always interacting with antigen-presenting cells that are presenting peptides. So it's almost like a test. Just imagine. Let's imagine you had to be identifying smells, for example, right? And this, right? So they would offer you smells, and I mean, if you're so sensitive that one smell makes you faint, they'll say, no, you're not good enough, right? So, and then you keep going. Just imagine such a game. It's exactly this. If you've got a receptor on your surface that's going to be not working, you are not going to be a good detective, right? Because you need your receptor to understand where the peptides are. On the other hand, if you're too good, you can be a bit dangerous because you can recognize household-derived peptides and will lead to autoimmune disease. And we are all aware of lupus, for example, right? So I would only want to say, and if you want to read, this is a beautiful paper, Ludger Klein, 2009. This orchestrated journey is about 2 weeks. A given cell has about 2 weeks to do this journey, and either it makes it or not. And it's shocking. It's an exquisitely stringent test. Less than 5% have sort of passed the test, right? Which is, that means that it needs to be very, very, very good job. How do they do it? As I've said, the interactions are always mediated by the T cell receptor talking to the peptides displayed on antigen-presenting cells. If your T cell receptor as a T cell doesn't get a signal, you die by neglect. Let's imagine, you are in this perfume shop and you don't smell a thing, don't buy perfume, right? Neglect. If you have too much, too sensitive to it, then you faint, can't do the job, out. But if you're intermediate and you recognize a range, you're good enough. Is that clear? 5% chance, which is, as I've said, very stringent. So where do we come into the picture? I have to say, for me, yes. Can you stand up and, please?
At what stage of human development is this thing happening when
very
a
good
baby?
question. Very good question. Yes. So it's happening. I know more about when it happens in mice, but this happens right about the final stages of embryo development. Very good question. And in humans, this is why children are so, very good question, because now this is forcing me to explain. This is why children are very vulnerable, because their immune system is developing. Thymic selection, this process is called thymic selection. It hasn't happened yet. All this latest stages of embryonic development and birth. Yeah, few weeks. So the mice, for example, that we were looking here were about 7-8 weeks. Well, not us, but our collaborators. So I'll explain that in a minute. So where can we now go as people who deal with equations? I mean, I would say that this is what I do. Make use of some equations. So as I said, there is a journey of changes. These cells come as stem cell-derived cells, and they change. biologically, a change is called differentiation. Let's not worry about calling things any names, but just imagine you can be in population 1, then you can change and become population 2, and then you can change and you actually have 2 choices. Do you like 4 or 8 more? And that's exactly what it is. You know, if you do something, then you become a 4, and if maybe you don't, you become an 8. This is the CD4, CD8 selection in the thymus. So just imagine you come from the bone marrow, you can be here. When you're here, you can die with a certain rate, mu1. You also can maybe differentiate, depending on what. I mean, I'm not drawing the interactions, but you see, this is my cartoon, you can see that. So I'm not drawing all these interactions, right? Put lots of antigen-presenting cells there, because they're mediating those transitions, but the idea is that at any stage, well, you come in from the bone marrow, you can die, or maybe you differentiate, you can die, you differentiate, and in these last 2 stages, you can also do something else, which is divide. And then you can exit. These 2 arrows here means that you exit the thymus. So this is the picture of the thymus, the equation version of the thymus, exiting to the blood. So what one can do is, if I introduce N1, the number of cells in this population, N2, the number of cells in this blue compartment, N4, the CD4s, and N8, can I use experimental data to understand? I'm only putting this not to scare anybody away. I am very sure there are people who love equations, and one shouldn't be embarrassed to show equations. I mean, I like equations. For my parents, that's a big problem, but you know, what to do? I like equations. So all these drawings can be incorporated in equations. And I'm just going to let's look at population 1. You come in from outside, it's like water coming into your container. You have one hole here and another hole of water going to compartment 2. How that is translated into an equation. So this population 1 is changing with time. That is, it's changing in time, the rate of change as a function of time. It goes up with the flux phi. Yes? Coming in, water coming into your container. And then you've got 2 minus, everybody knows this is a minus sign, 2 minus signs, lost terms. You lose one because of death, and that's just there. The other one is differentiation going into compartment 2, and you see that this term is also here for population 2. So that's the idea. Doesn't matter, yeah?
How do we know for sure that the rate is proportional to the amount of disappearance?
Very good question. We do not know. We hypothesize. But you know, I cannot go inside the mouse and look. We don't know, but you can imagine if, okay, you can make this assumption. That is, by the way, a very advanced question. You understand? So thank you for the question. What you can say is, per cell, each cell has a rate. And if all of them have a similar rate, because they're very similar, then it should be proportional to the number of cells.
Why not like N1 square?
Of course, because that is making life a bit harder. And this is, we are going to have a lovely time where I can write pencil and paper. But you could try. Yeah. No, that's a very good question, but we're not going to talk about model selection here for today. After the talk, if you wish, yeah. I mean, this is just... But it's good to have all questions, because that's... oh, yes. Actually, maybe... okay.
Last one.
Oh, yeah, you. Last for you.
From my side, yeah. Have you identified if the processes are Markovian?
Oh, Markovian. My collaborator, Martin, we can talk about Markovian
Sure.
at the end, please.
I think we'll Markovian part.
Yes,
after.
yes. And there was a question at the back?
Are the rates constant, or do they keep changing with different factors?
Could you repeat the question? Sorry, I can't.
Are the rates constant, or do they keep
Oh,
changing?
no. So for right now, the rates are constant. Yes, oh my God. You've done your homework before coming to this lecture. You read the paper. So what you can do, and this is true because in these mice, after the development is complete, what you have is a thymus in steady state. Steady state means that it's not changing with time. You can make each of these equations equal to 0 and pencil and paper, which I really like, with a big eraser, because I make lots of mistakes, you can write them. I am not writing them to show off. I'm writing them for the particular reason that you can see that the number of cells, this is the number of cells in population 1 in steady state, can be written as a function of the parameters. How much goes in divided by how much goes out. And why is this important? I had, I would say, the luxury and good fortune to work with Chris Hawquist, an amazing immunologist. She shared her data, and this is published, so it's okay. So what you imagine is you have sacrificed these animals with all these ethical permissions, and you can count because you have your flow cytometry, and you know which markers define the different populations. But of course, what you measure is cell numbers. And our question was, can we quantify these rates? Everybody knows a number is a number, 7, 8. But a rate is how many units of something per time. So where we had to get a sense of time. How did we get a sense of time? Some other people, and also her group, had measured on average, because of course this is all, you can, with labeling, actually with radioactive labeling, you can do that. You can quantify how long on average you wait in each of these compartments. So the waiting times in the different compartments are of the order of 60 hours, 16, 96, and 96. With that, you can do some mathematics, some statistics, and you can get rates. Forget about the rates. For me, the beauty of our exercise was here. Remember I had said this stringent test is around 5%? When we calculated with her data, and for mice, we got 9%. And we did a very basic mathematical statistical exercise. Right? And you can see that it's not that we had lots of mice, 8. So to me, this is sort of the adventure. You can do some math, and you can support the empirical observations that biologists working very hard are making. Let me just go quickly through this part. I've told you about two-photon microscopy. Two-photon microscopy allows you to see in vivo, in space and time, the interactions between, this is a lymph node. Imagine you have your antigen coming through your skin barrier, it breaks in, it comes in, dendritic cells, antigen-presenting cells talking to T cells in the lymph node. So there is a very good immunologist in Pasteur Institute, which is always great to visit, who works with two-photon microscopy, Philippe Bousseau. If you have a chance, read this paper, it's beautiful. So what is it that you can do? You label your cells fluorescently, you transfer them to your experimental animal, and you put the little animal asleep under the microscope. And you can take stacks of images, you can put them together and make a little movie. I didn't want to have too many movies, but you can find lots of movies with two-photon microscopy. And what you can count is cell contacts. As you can track all these images. So this is my theoretical description of these interactions. In red, and I'll just play this little thing, in red is the T cell going to your dendritic cell in green. We are theoretical scientists. We don't like funny shapes. So just imagine the dendrites of the dendritic cells are of length order B. So this is almost, and actually they're sweeping these arms, so you can almost imagine they're these spherical dendritic cells of radius B. And you've got your naive T cell at a distance R0 from that dendritic cell. So something that you can imagine is these are bigger cells. Dendritic cells are slightly bigger. They're not motile. These are smaller, small lymphocytes. The T cell, we are going to assume, and don't ask me why, and it's not quadratic. We're going to assume that my T cell in the lymph node is performing a random walk. We've got tiles here, so this is perfect. What is a random walk? I place myself here. And let's not be biased, right? I can move to the right, to the left, up and down, yeah, with the same probability. So you throw your die, right? You have your 4 options with the same probability. You throw, you should have done this exercise. You throw the die, right, front, yeah? So you keep moving like that, one little step at a time with these rules. Diffusivity D. Diffusivity D, it only tells you if I jump 1 or if I jump 2. In one step, right? I don't have strong legs, so I only jump 1. And I'm wearing heels, so no jumping. So then you can say, well, this is not happening in isolation. I've got my lymph node here of radius r. So I've got a spherical lymph node of radius r, my little antigen-presenting cell, the trajectory of my random walk. 3 numbers you need to remember. The size, B, of the dendritic cell, the radius of your lymph node, and the diffusivity, how fast you move. One can actually show that the mean time to find average over the sphere is this equation. R cubed should make us think of a volume. Yep. And this has units of time, which is very important. Yeah. So, and as I've said here, this is your Brownian motion inside the sphere for those who know too much, which is always very good, reflecting boundary conditions. Right when I get to the boundary of my lymph node, I reflect. And absorbing, because when I get to my target, I stick to it, my antigen-presenting cell. So what one can actually do is, we know that waiting for an event to happen is exponentially distributed. Have you heard of exponential? E? Just a mathematical function that we like. So we can use, if this is a time, the inverse of a time is a rate. We can use that rate to understand what is the rate at which these 2 encounters happen in the lymph node. So when we did that, what our immunologists wanted to answer was, okay, if I vaccinate, how many antigen-presenting cells do I need to have in the lymph node to make sure I trigger an immune response? So how did we help? So the rules of the game are, as I've said, ABCs are fixed, radius B, T cells move with Brownian motion, diffusivity D, my lymph node has radius D, and I've shown you, or one can show, that there is a characteristic time or rate associated with when these 2 come together. So now I can ask myself, well, in my lymph node, I not just have 1 antigen-presenting cell, I may have A of them, and I don't have just 1 T cell, I've got N. So what is then, this is important, the probability that out of the N T cells that I've got in my lymph node, one of them, at least one of them, with one of them doing the job, that should be good enough, hits any of the A during the time I'm imaging or during the time of my potential immune response, right? One can show that this is this expression, this probability 2 has that expression. So what the immunologist was asking himself, but he couldn't phrase it in mathematical terminology to make use of the data. He had the data, so we only did this little thing. So if you believe P2 is the probability that at least there's 1 encounter in that time, and of course we believe that it's an encounter with a specific antigen, it would lead to T cell activation. Let's imagine that you say, okay, let's imagine that probability is equal to 50, you know. Probability that it's every other time it happens. One can then show that you require 85 dendritic cells in your lymph node under the assumption that 1 in a million cells, T cells, carries the specificity required. Yeah? So, which actually is it's a very good number. I will finish with the final question that I put in the abstract for this talk, and I thought it would be important to answer the question, how can mathematics help you quantify the dynamics of a virus replication in vitro? Because with Ebola, as you know, and the people in Congo right now are suffering, right? The Ebola is pretty nasty, so it's not that in the lab it can just be dealt with. So I'm not going to talk too much about the details. This is, as I've said, a very nice review. You can look at it. Ebola has this little hook shape. It goes into the cells, and it does subvert lots of innate immune responses, so that's why it's so bad. Ebola is not so much terrible because of what it does, but because it delays immune responses, innate immune responses. So by the time adaptive immune responses come to you, it's rather late. Yeah. So we know it's very nasty. It's also very nasty because it can replicate in many cell types. And bad for the immune system because it can replicate in the cells that are very important. Macrophages, dendritic cells, endothelial cells, for example. It releases without killing the cell, so it's like producing, secreting out of the membrane. And as I've mentioned, It's a very dangerous pathogen. You need to only work with it in vitro in special biosafety level. And we had the fortune to have some data from that lab to work. And unfortunately, there's still no vaccine. There's only one which is currently on trials, only on compassionate grounds it's administered. So I'm not going to explain more than this. How can one make use of mathematics or equations? So what one can imagine is you've got your target cells, virus goes in, there is one period in which more virus can go in, and that's called the co-infection window. So this is this green period here. Then there is a longer period called the eclipse phase, which is the time which you've got an infected cell that is not making copies and getting them out. It might be making copies, but it's still not releasing de novo particles, so that's the eclipse phase. And then cells can move into what we call the productively infected cells, these eyes, until the cell die. But during any stage here, these cells can secrete the wild type of the virus, or viruses, you know, viruses mutate a lot. So when they are making use of the cellular machinery to replicate, they make mistakes. So some of these viruses are inert in the sense that they cannot infect. And for some viruses, you know, they make lots of mistakes, others are. So just in a nutshell, this is all I'll say about this. I will then conclude with looking forward. And as I was saying, the immune system has an important, and I would say very novel role in understanding responses to tumors. What has been observed is that there's better prognosis if near your tumor, the infiltration, we said immune cells are very motile, so when they move, when they migrate to the tumor, if they are of a certain type, like natural killer cells, then or CD8 T cells, there's better prognosis for the patient. I mean, I'm not going to, this is not set in stone, but there is evidence to suggest that depending on the profile of immune cells that migrate to the tumor site, you have a better prognosis. On the other hand, if you've got lots of ILCs and some funny macrophages, your prognosis is worse. So understanding which cells you've got can actually help understand not just treatment, but also stages. I wanted to add this to make the point even more. In a number of leukemias, cancers to do with blood cells, cancer immunotherapies are going to be very important because if you compare, and here are just examples, you can imagine, different cells interacting with different molecules, in these leukemias, those interactions are disruptive, changed. So understanding those changes might help. And there's nothing better than trying, sort of one of the advantages of these immune therapies are the fact that you're treating the patient with their own immune system, right? Which has lots of advantages. I will finish with another huge problem in some of these potential therapies. I don't know if you've heard, I've been telling you, all these CD8 T cells can kill infected cells, but in the cases of chronic infections, these cells get tired. In fact, they get exhausted. There's something called CD8 T cell exhaustion in chronic infections. So like HIV, for example, if you've got a virus for a long time, it's almost like these cells run out of steam. So understanding what are the molecular mechanisms that lead to that exhaustion is very important because it's also being seen in cancer. Cancer is something chronic, and it's being observed. So understanding the differences, understanding the molecular mechanisms of exhaustion in infection and cancer is essential. And maybe just before I finish, what is the difference, because I don't think I've explained, between an acute response. So an acute response is the virus is in dashed here, so goes up and down, but it's cleared, and your cells that can kill, if you've cleared the infection, you can get rid of them. In a chronic case, in a chronic environment, you never clear completely the virus. You see how you've got a little plateau here? So you need to maintain You maintain these CD8 T cells because you've not clear antigen, but they turn into the pink color, and they've decided pink is exhaustion. So pink phenotype, exhausted cells. So understanding, the differentiation pathways between fully active killing T cells and exhausted cells in cancer and infection is rather important at the minute. I will conclude with saying a huge thank you to the outreach team at ICTS, because it's been really wonderful. I really need to say thank you to Maria Nowicka, Mark Day, and John T. Carruthers, who did all this work. The immunological collaborators, Chris Hocquist, Susanna Celli, Philip Busso, Joe Gillard, Tom Loss, and Sophie Smither. Without them, I wouldn't be here today. We couldn't have done this work. Laura Liao, who actually is right now, she just left ICTS last night to go back to the US at Los Alamos National Lab. She's taught us a lot of virology. And a statistician at the University of Leeds, John Paul Gosling, Martin Lopez Garcia, who's been organizing a meeting at ICTS, and Grant Lythe at the University of Leeds. And I will say thank you to you for your patience, your questions. Before I conclude, I think it's important to be acknowledging the support we get with the grant income we get from DSTL, the Research Council in England, the European Union, at least for the time being, before Brexit or whatever, British Society for Immunology. Well, Suma knows, and I've got the pleasure to have a great scientist here with me. We had, a Spanish individual in the UK, with Suma and 5 other institutes in India, we had all this research exchange funded by the European Union. Isn't that beautiful? So we have to acknowledge that taxpayers' money is supporting us to be here. The British Society for Immunology and the University of Leeds. I will conclude with saying, if you're interested to learn more, we coordinate one of these European networks to train PhD students. It's been running for a year. It's called Quantitative T-Cell Immunology and Immunotherapy. We'll host an event in Leeds and more events to come in the next 3 years. And if you've got a PhD in maths or physics and you would like to come to work with us, there is a job at the minute. With this, I'll finish and take any questions. Thank you very much.
Questions?
The shape of the protein, the 9 peptides, the shape, the structure, is it in a folded state, native folded state? How is it? Is it that shape important, or it's just a sequence?
Well, I don't know much, but I think it might, okay, it might be important. It might be important in as much as it fits. Just imagine, right? The T-cell receptor, it's almost like a molecule with arms.
Right.
So the peptide has to be sort of in one particular fold.
Yes.
But it's not lock and key. I mean,
No, that's
just
okay. I mean, it's kind of fuzzy. But in the sense that, is it the shape? Because there is no other way of,
okay.
Or is it mass?
Okay, let me tell you something. Something that shocked me is, whereas for B-cell receptors and the antibody recognition, that seems to be, so when 2 things come and bind, there is a change in the energy and in the entropy. Are these things familiar to you? Okay, because I thought you would. So there is a change in energy and entropy as one would expect. The greater affinity, those changes support that. For T-cell recognition, none of that intuition that we may have built about energy changes and entropy changes are the case. So in as much as shape matters, it's not the only thing. And we still don't know, or I don't. Yes, at the back. Thank you. She was first.
Hello. No one's asking, I'll ask.
Ah, there.
Typically, as a layman, I know that stem cells are harvested from the bone marrow and the umbilical cord of newborn children. Can we harvest T cells artificially in the lab?
Sorry, I didn't hear the last.
Can we harvest or grow stem cells artificially in the lab?
So, for example, I didn't mention here, but some of these if someone wants to replicate in vitro the thymus development, that's exactly what they do. They take cells from the bone marrow and they culture very carefully, of course. But stem cells are very difficult to work with because they're very, I don't know how to describe them, but basically you just need to know very well, and they don't like lots of things. So keeping them alive is a struggle, but yes, that's one way.
So can we use stem cells from other species to test on human organ?
Well, that would have lots of issues because of this MHC recognition. So this molecule I've mentioned, that antigen-presenting cells present the peptide, that needs to be specific to us humans. And that's, okay, now that he's asking me, I should say this MHC molecule is the one that brings us all the problems in transplants. So even within individuals, that already causes transplant rejection, so let alone going to other species for those purposes. Yep.
Thank you.
Thank you.
Have you considered TCR recycling when you do your calculations for the germinal center, or how many dendritic cells are needed?
So that's a very good question, and yes, I mean, I would say there is, so when T cells signal, they internalize, but I thought for the talk today that would be, I don't know, too much intracellular machinery, but yes, you have to, something cells do very regularly is, since they've already received the signal, they internalize all the receptors, some of them recycle, some of them are degraded, so there's all this trafficking. So yes, one, not just for T cells, but for lots of receptors, absolutely.
Hello. First of all, compliments for your wonderful presentation.
Thank you.
I have basically two questions. The first one, before you conclude, there was a prognosis about the tumors, where you mentioned a source of that West et al. 2015. In that, as you say that, the prognosis is grave if there is the antigen presenting, yes. Mm-hmm. When there is the CD8 and NK cells, you say prognosis is better. Mm-hmm. But in the next slide, you said that the prognosis is poor when that kind of cells are not there. Exactly. So, as a mathematician, have you any sort of such algorithms or mathematical models that can, illustratively explain this kind of a flow, that exactly this is happening or not, or at least if we can predict it?
Well.
One, and the second thing,
okay, that's fine.
Just a couple of days back, there was a report in New York Times that there is a receptor, that's CD47, that actually CD47, okay, that actually masks the cells from getting destroyed by the immune system. That is with most of the cells. Now, this cancer cells are very cunning. They often get this receptor imbibed out of some sort of mutations which is still not understood. And when they have this CD47 on them, they are not recognized by even the immunotherapy, whatever is rendered for killing those cells, and then they escape out. So, as a mathematician and all the models that you explained, could that be any solution or potential, approach towards that? That's all. Thank you.
Right. So the answer to your first question, and do I have them here? I may do, may not. Let me just see. Some of the cells I didn't tell you about today, but I think I've seen them here. Regulatory cells, Martin? Yes, here. So there is another type of cells. I didn't talk about them today because then this is a zoo, and we don't even, clear ideas is always important. So there are a different type of cells called T regulatory cells, and as the name suggests, they regulate something. So what they regulate is too much cytotoxic activity. Let's not go into the molecular mechanisms, but yes, indeed, one student who finished last year worked on understanding the balance between how many regulatory cells you've got in your tumor and how many CD8 T cells you've got. And something that we still don't even know, so let alone whether, what matters is the absolute numbers of these cells, how many cytotoxic and how many regulatory you've got, or if it's the ratio. If it's only the ratio that matters, or if it's a matter of absolute numbers. Back to what you said about your CD47, I don't know that molecule, but I know there's another molecule called CTLA4, and PD-1 and PD-L1 that tumors are able to play around with to really make things bad for the patient. So, and it's called checkpoint blockades. And a lot of people, in fact, what happens is a lot of people in some immunotherapies, they develop those sort of blocks and the therapy stops working, absolutely. I mean, I have to say, I wouldn't say cancer cells are canny. They're just different. Right? I mean, cells don't know. I mean, I still remember, it's like, oh, how does the cell know? Well, the cell doesn't know. The cell has molecules that are subject to some rules, and they do what they do. So cancer cells are our own cells that are dysregulated, and they just have advantages. No, I'm just being very silly, but I would just say canny. Why? Because exactly. Well, yes, but you see, the mutations. I mean, this is something I'm not talking about today. A lot of these mutations are induced by the therapies. So it's just you're forcing the system. So it's almost like, let me draw, because so this is your cell, and it has some receptor to see this is your cell, and it needs to bind to something. Then you add your therapy, and you're changing something. So you know what happens? Since what you're blocking with your therapy, maybe you're blocking this so that it can't bind, the cell needs to explore. They're curious. So they upregulate another receptor of a different type. So the therapy is forcing those mutations. Those are well known. So it's almost, like if someone's pushing you the wrong way, you're going to get angry. Sorry, I think other people have a fair, yeah? Yeah, well, later. Let's go in turns.
Entirety of your calculations, was there a role of charge to play with the rate of cell division or whatever was to be produced? Was there a role for charge in this? Charge of the particle?
So here you're talking about these numbers? Are you talking about this part? Sorry.
The initial derivation which you had.
The initial derivation?
Initial equation which you had prepared. So was there a role for charge in the rate at which everything took place?
Here? Oh, so what we did,
in your observation.
So so the way we worked was, what you have a sense is you have a sense of, for example, I mentioned residence times.
Sorry, can I understand? I think she's asking whether charge. You mean
charge of the particle or the
charge
particular cell? Did that have an influence in your
the
equation?
charge, the electrical charge.
Yeah.
The electrical charge, you mean?
Charge of the particle.
Well, that's a very good question. Electrostatic charges are very important, but I would say it's ion fluxes and things like that regulate. So yes, but for this, I'm sure it matters, like for example, for the protein structure, but talk to Suma. She needs to do another KAAPI. Lots of coffee here, lots of curiosity.
Question over there.
Ma'am, I have one question.
Yes.
Do you have a mathematical model which tells that how much memory cell will be produced from how much infection load in case of B cell?
Well, the man is there. Well, I mean, but yes, there are lots of mathematical models. There's actually a lovely group in Emory, Atlanta, Rustomantia. He's actually from India. Well, there you are, lots of great Indian scientists. So yes, how the viral load will determine your immune response, absolutely. But I don't work on that.
I have 3 questions, if that's okay.
Sorry, maybe one.
Okay, one. Okay, so
over the coffee you can ask them.
Okay, so when you were talking about Ebola, you were talking about mutations.
Yes.
How significant is the rate of mutation, and how does it tie onto the whole thing?
Oh, you get me there, Ebola. I mean, I don't think Ebola is particularly, just standard. Nothing to worry about, this standard thing of a virus. So I think for me, the understanding of how dangerous Ebola is much more how it subverts the antigen presentation. So as I was mentioning, so not so much the mutation rate as such, but the fact that it infects macrophages and dendritic cells. So there's 2 dangers with that. These are very motile cells, so there's going to be lots of dissemination if they make copies, but then they subvert this antigen presentation. So they delay the talking to the T cells. So to me, that is why Ebola is so nasty. Yep.
How far away are we from vaccination for the common cold?
Vaccination from?
For the common cold, or any cure for the common cold?
For the common cold, you said?
Still don't have a cure for common cold, right?
I mean, I know very little. I mean, most vaccines are sort of B-cell vaccines, and I know zero about B-cells. So I think you talk to the director of this institute and you suggest you bring someone. But, I mean, how far? I mean, one thing is, even if we are identifying some mechanisms, something that I'm becoming acutely aware is the time to make a scientific discovery into clinical practice through the clinical trials, it's a very long journey. But, you know, it's good to keep thinking about things, so yeah.
Maybe one last question and then we can break for coffee.
I have one question. Does this process has any relation with Fibonacci series?
Sorry, slowly because there's some echo here. I can't.
Now it is okay.
The question?
Does this process has any relation with Fibonacci series?
Which process?
This cell mutation.
Clonal selection with?
Fibonacci series.
Sorry.
Fibonacci.
Oh, Fibonacci. Oof. No, I don't think so. No, I mean, this is only, just imagine. I mean, I think with clonal selection, something that I've said, and I'll draw here again. So if this is your T cell, and this is your antigen-presenting cell, right? Let's imagine you've got your antigen-presenting cell, MHC here, peptide there, T cell receptor there. Before you're even getting there, it is a matter of, just imagine, have you ever traveled in a crowded bus? Someone tells you, find the person with the red coat. So depending on how crowded, so already, even regardless of how good as an antigen-presenting cell or how good as a T cell, you need to find each other. And the lymph node is a crowded environment, number one. As I was saying, the interaction there, let me tell you, one T cell receptor can bind 1 million different MHC peptide complexes. That's a lot. And one MHC can bind, so as I said, cross-reactivity and promiscuous. So what usually happens is what determines the first expansion is a matter of who gets there first. Yeah? And sometimes it might be that it's a T cell receptor that is not the fittest, if we want to call it, but it's just doing the job. So the idea is that sometimes it's not a matter of being the fittest, but being there early enough. You start controlling the virus, and then you buy time to then find maybe more rare cells. Does that answer your question?
Okay, let's thank Caroline again, and I guess we can have more questions over coffee.
Thank you.